The shared resource provides advice and guidance during the development of new protocols, where investigators are interested in pharmacologic investigations, dosing schedules, dosing intensity, development individualized dosing, or regimen personalization.
Designing pharmacokinetic, pharmacodynamic, and pharmacogenetic methods and aims, development of grant applications, drug or biomarker assay method development and validation, coordination with the clinical trials office regarding the collection of the human specimens and transport to our laboratory, storage, analysis, and running of the research subject samples, pharmacokinetic, pharmacodynamic, and pharmacogenetic modeling, selection of genetic markers for pharmacogenetic studies, data interpretation, publication, and manuscript preparation.
In this project, we're studying how patients who have NF1 syndrome will metabolize drugs. NF1 syndrome is an inherited cancer predisposition syndrome. And we're creating a porcine or swine model of NF1 syndrome. We're going to be testing drugs that are used in NF1 patients or might be used in NF1 patients to determine how they're metabolized in wild type or pigs that have the introduced NF1 mutation. Measuring the concentrations of drugs over time in an animal is something that we can't do. We don't have that skill in our own lab, and the clinical pharmacology shared resource has the expertise to measure these drugs over time, determine the area under the curve, and look for changes in the profile of drug metabolisms. And these are techniques and involves techniques we don't do and expertise we don't have.
The lab's role in this is to assess drug pharmacokinetics, which means drug exposure, looking at variables like drug metabolism and looking at these aspects to figure out how we can best decide on an appropriate dose for the animals and then translate that up into humans. We have expertise in measuring drug pharmacokinetics, and so we have instrumentation such as tandem mass spectrometry. We can take these timed measurements and then we can essentially draw a curve through those dots or those points, those concentrations, and get what's called an area under the curve. And this in turn gives us an idea of what sort of exposure that the animals are having depending on the dose, and it tells us, it guides us as well, do we need to increase the dose or decrease it? Do we need to shorten the frequency of administration or keep it the same using parameters like drug half-life?
We have never done any sort of pharmacology studies or drug studies up to this point in pigs. And so we really needed some expertise in the design and development of how we do these experiments, including how many samples we need, what the controls are going to be, and the methods used. And so the clinical pharmacology core was really helpful in sitting down with us ahead of time before we even started experiments and really planning them out, and detailing the specific methods that they would use to get the data that we were looking for.